Hey everybody, welcome back to Microbiology. We're going to go through the continuation of antibiotics, and this is going to be focusing on antimetabolites. So sulfonamides and trimethoprim are going to compete with either PABA or dihydrofolate reductase, and all of this is just to prevent making the tetrahydrofolate, which is the precursor to the purines, and of course purines make the DNA. So these have to be given together for the most effective effect, right? So because these two are synergistic, and they are going to be broad spectrum bacteriostatic, and they are given to treat UTIs, RTIs, GIT infections, and the resistant mechanisms are going to be efflux, alteration of the enzyme, overproduction of either PABA or DHFA, folic acid dependency, or decreased permeability. So in these bacteria, usually the bacteria have to make the folic acid. That's the point of the PABA, and that's the point of DHFA. But those who rely on the folic acid from the host cell, they can actually just use the host cell's folic acid, make the purines and the bacterial DNA. So there is not going to be any real use for the sulfonamide and trimethoprims. Dapsone paraaminosalicylic acids are going to be similar in mechanism. They're going to prevent the formation of folic acid. However, they are very, very particular. They're specific to only Mycobacterium. Same thing with the sulfonamide resistance. There's going to be efflux or folic acid dependency. This, however, is going to be given as a topical gel. So the sulfa drugs, sulfonamide and dapsone, are going to work up here to inhibit like the first step of the folic acid synthesis, and then trimethoprim is going to be like towards the end. That's why we had to give them together. We can't give them to anybody with sulfa allergy. Okay, so the DNA inhibitors are metronidazole quinolones and fluoroquinolones. The RNA inhibitor is only rifampin. The resistance mechanism to these two are going to be alteration of the target site, so these aren't going to be able to, bind to the DNA. Decreased permeability and increased export. Metronidazole is going to be a bactericidal. It's pretty narrow spectrum, and it works against anaerobes and protozoa. The quinolones or the fluoroquinolones are going to be ciprofloxacin, ogalocin, and norfloxacin. Cipro is the one that you use the most in the hospital. These inhibit the DNA topoisomerase and the topoisomerase 4, so 2 and 4. These are going to prevent the supercoiling from unwinding. So it's going to coil, and then the DNA is going to break. So it is a bactericidal. It has a broad spectrum. It's good against gram-negative, gram-positive, anaerobes, aerobes, and is given through an oral route. Rifampin is going to be, or rifamycin is going to be the main RNA inhibitor. So it is going to have rifamycin, rifampicin, and rifabutin as its derivatives. It inhibits the DNA-dependent RNA polymerase, and by doing that, stops the RNA synthesis. It's pretty good. It's bactericidal, broad spectrum. It works against gram-positive, gram-negative bacteria, along with the Mycobacterium. The problem is this can be easily resisted because, remember, RNA is prone to mutations, and that's why the bacteria can evolve so fast. Oh wait, important. This is good for pregnancy, so this is not going to cross the blood-brain barrier. It's okay for pregnancies. The antimicrobial susceptible testing. So we first use the symptomatic diagnosis, and then we give the empiric therapy based on the diagnosis. And then after the tests come back, then you can give them or the patient's specific therapy. So how do we find the specific therapy? We use diffusion test, dilution test, and a whole lot of other testing. Okay, so the diffusion test. We use the zone methods, really. So the Kirby-Bauer method is to test for antibiotic sensitivity. You put the antibiotic a disc onto an inoculated agar plate. Allow the disc to be, equidistant from each other. Allow the bacteria to grow, and you want to see the diameter of each zone of inhibition with a ruler. So this is going to compare which antibiotic is more effective for the bacteria. So we want to see if they're susceptible, if they're intermediate, or if they're resistant to the antibiotic. This is not a qualitative method, so it's either like these three, right? Or a quantitative method, sorry. So it's based basically on the technician or the pathologist's experience, whether they're able to see like minor changes in color or zone size. So this is not a very, very good test, but it's fast. The Stokes method is kind of the same thing, the antibiotic sensitivity test, and you want to see the antibiotic effectiveness against different pathogens. So for this, you're using different kinds of antibiotics. For this, you're using different pathogens. Once again, it's kind of the same deal. You want to compare the zone, the inoculated plate, antibiotic on top of this, and then you wait for the zones to form. The E-test is going to test the effectiveness of the antibiotic, and this is the only one that's quantitative. So it measures the minimum inhibitory concentration. So this is done using like titers. Okay, so the dilution test, this is quantitative. They're going to measure the minimal inhibitory concentration and the minimal bactericidal concentration. So you want to see when the red color is clear, because red color, that means the growth of the bacteria. So you want to do a serial amount of dilutions to find the minimal effective antibiotic concentration. The MIC value is going to be only the bactericidal concentration. The MIC is minimal bactericidal concentration, and it's going to be the concentration of the drug that kills at least 99.9% of the pathogen. So why is this important, right? Antibiotics are fairly toxic. We want to give the least amount of the drug to the patient to minimize the side effects, and yet we want to make it we still want it to be effective. So we want to see just the bare minimum that we can give. Automated tests are pretty good. They are going to be fast. They use a Vitek 2 system, which is a whole lot more in depth than any of you guys really want to go into, unless you're going to be a researcher. But as anybody with who's going into clinical practice, we just write it down. We want this test, and we send it off to the lab with the samples. That's it. More useful are the phenotype tests. So we're going to do a spot test, which is a rapid colorimetric assay to detect beta-lactamases. One of them is the NitroCef test. So each disc is going to be soaked in NitroCef, and on the incubation of the disc with E. coli, we're going to see if a red product is formed. Once a red product is formed, that means it's a positive result. That means this patient is infected with good old E. coli. It is pretty fast. You can get a result within 10 minutes. The chloramphenicol, remember that is an antibiotic. Modifying enzyme is chloramphenicol acetyltransferase is going to be even faster. And by that, we put the chloramphenicol onto an inoculated plate, and then see how big the zone of death is. And it's pretty fast. Genotype test is going to detect the resistant gene VAN A and VAN B for vancomycin. So we want to see if any of these bacteria are going to be resistant. It's rapid and it can detect the presence of the resistant gene, but it doesn't actually mean that it's going to it doesn't necessarily mean that it's going to detect the expressed gene. So this is not too useful as a test to see if the antibiotic is viable. This is more like, did this bacteria mutate or not? That's it for now. Thank you for your attention and good luck studying.